It is my pleasure to introduce Leor Weinberger from UCSF. Leor did his undergrad at Maryland, then moved on to do a PhD at Berkeley, and then went on to do a postdoc, a fellow, a Lewis Thomas, as it was called before. I think now it's called Lewis Sigler Fellows, but an independent fellow position at Princeton, where he worked with David Botstein and Tom Silavi, and then went on to start his own lab at San Diego, UC San Diego, before moving on to UCSF, where he has spent the last couple of decades doing really exceptional work. So, initially I thought I will just go over his CV and say all the nice things. So he has gotten a lot of awards, Pew, DP2, which is New Innovator, and then Transformative RO1, and several other long list of awards. But I think the one that really stood out for me, which I think for a mentor is a very satisfying one, is in 2018, he got the Outstanding Mentor Award, where your former trainees write letters for you to nominate you to be a good mentor. So I think that speaks volumes to not just his science, but also how he treats people and think about their development. So that's to me, that's very exciting and very amazing. I also wanted to bring up how, usually there's almost like a dichotomy, at least theoretically. People say you either do, creative or um, abstract work, or you do, work with applications and implications. I think Leor is one of the few people where you really go from really creative, very unique systems, and using noise and feedback methods uh, at the very basic level, and then he goes on to implement it with real translational potential. So that's a very unique aspect of it, and I think it's very inspiring, and also the interdisciplinary nature of it. This institute really started with the idea of combining mathematics and biology, and you will see a lot of what Leor does is informed by mathematics and stochastic systems. And for me, so my lab now is doing some viral work, but even before that, his papers were really inspiring to me because they're so conceptual that you can remove the word HIV or viral infections and it still does the same job. And now that we are working quite a lot in this area, we have been reading all of his papers very carefully. So just a big fan of his work and very excited to welcome him to Northwestern. I'll just make one last announcement. And this is in your CV, so I think it's public. So Leor is now starting his own big department at University of Miami in systems biology, which is a very exciting new opportunity, which he starts in a couple of months. So all the best for that, and very excited to welcome you here.
Thank you. I'm grateful. Thank you for the invitation. It's exciting to be here. And I thought I'd start with a chalk version and explain where this idea came from as a graduate student. It started with a mathematical model when I was a graduate student, and I thought I'd go through that with you first and then take you through the talk. Okay, so I had been studying HIV even before grad school, working with Alan Perelson. And when HIV infects a patient or an individual, this is the viral load, the number of viral copies per milliliter of blood. The virus rises up in about 2 to 4 weeks to a peak, comes off of this peak, and comes to a set point or steady state load called the set point. It's typically around 10,000 to 100,000 copies per milliliter of blood. And that can last for 8 to 10 years, right? Until a patient, well, we don't actually know why. In the absence of therapy, these patients can last 8 to 10 years before the virus takes off, and that leads to AIDS, okay? And the level of the viral load, the set point level, determines how quickly this AIDS region is reached, okay? So the lower the viral load, the longer it takes. The higher the viral load, the shorter it takes. Okay? In some cases, it can be 2 years, very quick. And this is essentially what the drugs do, lower the viral load. I'll just remove this guy. When the drugs were introduced in the early 90s, what they did is the viral load would drop precipitously below the limit of detection. And at first, physicians and clinicians thought patients were cured. They would keep patients on drugs for long periods of time, in some cases decades, see very little, maybe some blips here and there. But these drugs called antiretroviral therapy, or ART, in some cases, highly active antiretroviral therapy. If a patient was taken off antiretroviral therapy, ART stopped, right? This is start ART, this is stop ART, right? Discontinue. What would happen is the virus would rebound to pre-treatment levels, to pre-steady state levels. And this was a big question. Where was this coming from? First I want to tell you about the model that explained this steady state, and then I'll tell you about ART and the other aspects. So a very simple mathematical model. What's that? Yep.
Do they speak? Do they see the background?
Okay. So there was a very simple mathematical model that could describe all of this data that was developed by Alan Perelson and in parallel by Martin Novak. And it was really just three compartments. The target cells, the CD4 T cells, which HIV infects. These cells are created in the thymus at some rate. They have a lifetime. We know that lifetime. They can be infected by HIV, one called HIV-X. This is the virus. HIV infects those cells, turns them into an infected cell, productively infected cell, which makes lots of this virus. And as that cell dies, it produces more virus. And this population of virus like this. This was the mathematical model. Very simple. This virus has a clearance rate from the body. These cells have a burst size, meaning every time they die, they produce N virions. We know that we knew on average that number. And these viruses can infect cells with some rate, which we still don't actually know that rate. Leads to three very simple equations, which I usually don't write down, but for this crowd, I will. Very simple coupled nonlinear differential equations. The target cells are created at a rate, constant rate delta, lambda. They decay, and then they're infected at a per capita mass action rate. The infected cells really just come from this same, and they die at a rate delta that I showed you up there. And the virus compartment is created every time a cell dies. You get N copies and cleared at a rate C. This simple set of differential equations, which took about, I don't know, 5 years to develop, fit all of the patient data that existed that I showed you here. What it didn't fit was that data when you remove from ART. And the reason it didn't fit that data is because there was this population, it turns out, of cells called latently infected, which were just essentially a copy of the virus quiescently sitting there. And very occasionally, these cells could turn on. The drugs are attacking only the actively replicating virus. This long-lived population could be maintained. And even if you had patients on drugs for decades, these cells could reactivate once the drug are spontaneously reactivating. And once the drugs are removed, they can repopulate this population very quickly. And that leads to pretreatment levels. So this was the problem. And the idea was, could we, instead of trying to cure the infection, these are very hard to get rid of, could we, in essence, put all of the virus into a population like this? And then the patient wouldn't be cured, but it would be a chronic, long-lived syndrome where all of the virus was in a silent or quiescent population, it wouldn't really be a problem. The patient wouldn't be cured, but the virus wouldn't be causing disease and AIDS. Could we do that? And the idea was, well, we could do it, but it would be the same thing. We'd be taking drugs continually. You'd have to give a patient drugs to push all the virus into this state. Maybe there'd be a way instead to give a single administration of something that would continually push the virus into a state like this, into quiescent state. And that was the kernel, the beginnings of this idea, which I'll now show you. So I'm going to get rid of this for a second. I think I'm going to redraw this. I ran out of a little bit of room. Okay, so we had the target population of cells here. By the way, this is called the basic model. Of viral dynamics. And now I'll write the other model here. So of course, it's production and decay that we had before. The virus X infects these cells, makes these infected or productively infected cells, which make more virus. So the idea was, could we make a DUD or a molecular parasite virus that was essentially a therapeutic virus? I'm going to call it check, okay? Instead of X. This check would infect the same cells, the target cells over here, and instead of turning them into productively infected cells, would turn them, you can't see this, can you? It would turn them into these therapeutically transduced cells. Let's call that, it's a target cell that's transduced. These would not do anything, right? They would just sit there. But they would be infectable. Oops, that looks like a check. It's not supposed to be a check. They'd be infectable by the wild-type virus. And when the wild-type virus infects those cells, instead of turning them into these cells, it would turn them into these productively infected cells that produced more of this therapeutic virus, this check. And so you get more of this therapy and very little of this X. And when we built this model and ran the simulations, essentially what this population here, IT, is a therapeutically transduced population that's producing the therapeutic candidate, therapeutic virus. When we ran these simulations, what we would get would be a new steady state that would be very low right here, right? This is, we called these guys TIPs, therapeutic interfering particles. And when we delivered the tip, we would get this new steady state that was very low. And the idea was it would reduce viral load. The patient, in principle, would take much, much longer to develop AIDS, maybe indefinite, and would have a much lower probability of transmitting the virus. This was the beginning 20 or more years ago of this idea, which I'll show you is now entering clinical trials. So it took us 20 years. That paper was the 2003 paper that I published as a graduate student. And just last year, we reported that we developed this therapeutic candidate, and it showed it is effective in non-human primates and monkeys. And I've started this nonprofit institute to serve as the IND sponsor to run the clinical trial. So it took 20 years. It was I would fully admit, a counterculture idea for most of that time. It was, to give you an idea, my greatest champions, the people who write me letters, who have, very well-known virologists who have prizes, would call this a holy grail of virology. And I'm convinced they meant that cynically. Like, this doesn't exist. You're never going to find it. Stop looking for it. So I'll show you it does exist. They are therapeutic, and we are going into clinical trials.
Yeah. So essentially, you basically generate a competitor.
A molecular parasite, not a competitor virus, because a competitor virus you'd get the same problem, right? If this virus, this tip, the check, could replicate on its own, then you would just have competitive exclusion. you would have it outcompete the wild-type virus completely, and you'd have a new problem, which would be the new competitor virus. This is a molecular parasite. It's a parasite of a parasite. Yeah.
Again, competing for the source of growth.
It's an intracellular, yes, molecular parasite. It competes for the sources is exactly what it does.
But it still kills.
It still kills. It still kills the cells.
So you will have this issue of decreasing the immune cells. Not
These ones.
Chad,
Right. These ones, not these ones. Right?
they don't kill.
These cells are not killed. These cells are just transduced, and they're completely healthy as far as we can tell, as I'll show you. Yeah. The monkeys live longer, dramatically longer. Okay, so this is the idea that Ertugrul pointed out. These tip RNAs are essentially competitor or molecular parasite RNAs that compete with the wild-type RNAs for packaging material, as I'll go through, and you create these dud or non-pathogenic viral particles that can transduce the next cell. This was the entire concept. It's a biocontrol theory. Okay. Now I'll go away from the board to this. Okay. Any questions? No? Okay. So this took a long time. I thought I would quickly put this in the context of the other projects in the lab, like 2 minutes, just so you understand where this sits, because a number of people yesterday said, are you going to speak about stochastics and noise or this other stuff? And so I thought I'd just give you an idea of the mapping out of the lab. I think our bread and butter has always been what Yogesh pointed to, the pathways that control noise, or these dither pathways. We discovered one of them, the first one in HIV latency, and then in embryonic stem cells a couple of years ago. And we essentially believe these pathways are acting like Bunsen burners for cells. They're amplifying stochastic fluctuations like Bunsen burners amplify thermal fluctuations in order to potentiate reactions, right? Accelerate and potentiate transitions. We then, in the context of studying these dither pathways, we discovered a set of feedback loops in other viruses. I'm not going to talk about that system. That's also a therapeutic candidate that we're working on. The idea for this second feedback disruption candidate is that we wanted to develop therapeutics that had a high barrier to resistance. And one way to do that is to target feedback loops, because when you target a feedback loop, the virus, in order to escape, has to mutate both the protein and, say, the DNA binding site. That's very difficult to do. Not impossible, but difficult. And that got us part of the way of where we wanted to go, which was therapeutics that are resistance proof, that have not just a high barrier to resistance, but an infinitely high barrier to resistance. How do you do that? Well, you have to make a therapeutic that replicates. You have to meet the virus. The virus is constantly mutating. The only way to have an infinitely high barrier is to have a therapeutic that itself can replicate and mutate. And that's what I'll tell you about. That's what these tips are. Okay. So I'm going to give a long introduction. I apologize. Yogesh mentioned this. This is my new home, University of Miami, starting a new department of Cell and Systems Biology. And we have lots of open FTEs and a fellows program that'll be announced. So please, send me your best. Okay. This concept, it turns out, that I worked on as a graduate student, that I developed in isolation. I was naive and ignorant of the fact that there was a precedent for it in the biological literature. And that precedent is now over 70 years old. It starts with this gentleman, Preben von Magnus, who was a Danish microbiologist who discovered monkeypox, but is better known in virology circles for this effect on the right that he lent his name to, called the von Magnus effect. When von Magnus and colleagues were studying influenza viruses in culture, they saw that shortly after they inoculated the culture, the viral titers, the amount of virus would rise to very high levels, peak, and then drop. And then it would recover and continue in this oscillatory type pattern, right? And this wasn't due to target cells. They would consistently give cells. So it wasn't that the cells were being killed off and causing this effect. And von Magnus predicted or hypothesized that this was due to what he called an incomplete virus, okay, that was the wrong key, or an incomplete virus that was essentially acting as a molecular parasite. This incomplete virus, shortly after inoculation of the culture, would spontaneously form, he believed. When the virus got to very high levels, that incomplete parasite would feed off of it, stealing from it and knock the viral levels down. But when the viral levels got to very low levels, there was nothing left to feed off of it, right? So the wild type would recover. He called these incomplete viruses. They're essentially a predator-prey reaction, right? That's what you're seeing. The incomplete is a predator of the wild type virus, which is the prey. Others called them von Magnus particles, probably in jest, because he had no evidence they existed. Maybe a little bit of indirect ultracentrifugation data, but no one believed these existed. And it took almost 20 years for the first evidence to arise. And that was with a microscopist, Adeline Hackett, at the NIH in Berkeley, who was studying a different set of viruses, VSV, vesicular ptomatitis virus. They form these stereotyped bullet-shaped particles. And here's the key of uniform length. They're always the same length. But when Hackett looked at these oscillating von Magnus cultures, she saw these short, stubby particles at arrow number 1, 2, for example. These were the incomplete virus. They had a mutation in capsid, and it was the first evidence of an actual incomplete virus. Unfortunately, everyone forgot about Hackett, and what they remember is this paper from this very influential American virology group, where they renamed these particles, defective interfering particles, or DI particles, or DIPs. And here's the important part, implicated them in disease processes. It was a very carefully worded paper, I think. It didn't say whether these were attenuating disease processes or promulgating disease processes or amplifying disease processes, implicated in disease processes. Nevertheless, because of the notoriety of the group and the temporal proximity to the prize, it's created a lot of interest, and many, if not most, virology groups started studying defective interfering particles, trying to isolate them, trying to build them as therapeutics throughout the 1970s. It was very popular. Unfortunately, this was irrational exuberance. The technology didn't exist to isolate these particles. So you had these mixed populations. You cannot build a therapeutic with a mixed population. You're going to give someone a virus. No one really knew whether they were attenuating disease processes. They could be, you were giving all these particles, it might cause some sort of cytokine storm and cause a bigger problem in the host. So that implication problem, there were many problems with this. And because of that, after the early 1980s, the field fell flat on its back and these were essentially abandoned, except in some laboratories as tools. But as therapeutics, abandoned. You couldn't find a paper or patent after the early 1980s on this. If you took a virology course in the 80s or 90s or early 2000s,
I did.
you did.
I did Baltimore.
You may have heard about these then.
He taught us about
Yeah.
DIPs.
Right.
His feeling about them was that they were basically the cause of the decline, so they attenuated the infection. But that was it, you know. I took that course and I was in a virology lab as a graduate student, sort of, and they didn't work on it any further.
Yeah.
So it got abandoned.
Yeah.
I completely forgot about it until you just mentioned it.
You're one of the rare, because you took a course with David, you're one of the rare people who knows about these. Most virologists don't even know about them. I mean, I took a course in the late 1990s, and 10 minutes of an entire semester, they mentioned these. And I don't think, whenever I mentioned the virologists, they're like, oh yeah, I maybe remember a little bit. But they were a backwater. They were described to me as a backwater. This began to change, and I wish this was more romantic, but it's not. It began to change in the early 2000s when this series of mathematical models that we put out got the attention of funders. And these funders put capital into the field, and everyone got excited again because there was money. So not romantic. And that's what I'll tell you about. Okay. Outline, I will tell you about the theory of a TIP, a therapeutic interfering particle, why we renamed it, or I would say renamed it, right, from a von Magnus particle to a dip to a TIP. These are a subset of defective interfering particles with an R naught greater than 1, an R0 greater than 1. And that essentially means that this arrow has to be larger than this arrow. This dotted arrow.
Do you also get oscillations in your system?
Good question. If you build it with an R naught greater than 1, you do not. They're damped. You can get damped oscillations, but not.
You can see those damping.
You can see those damping happen. Yeah, yeah. It's parameter dependent. It's R naught dependent. But yeah. I'll tell you about the technology that we have to develop over 20 years in order to identify and build these tips. First for HIV and also for Zika and SARS, and then the discovery of the therapeutic effect that they're not inducing some cytokine storm and causing more sickness, but actually attenuating disease. All right, and I give long introductions, so even though we're half an hour in, if you don't see a piece of data in the next 10 minutes, don't be alarmed, it will come. How I give my talks. A lot of people over 20 years contributed to this. I want to point out Fatima, who was the first author on the paper last year, and the co-first author, this is a long latency time, Elizabeth Tanner. They did the majority of the work, as I'll show you. Nancy was our non-human primate collaborator at Oregon. Davey is our clinical protocol chair at UCSD for our clinical trial, and then our FDA consultants, Joy and Phil, who helped, and of course the funders. And disclosures, I started a few biotech companies as well as the nonprofit that I mentioned to build these and various other compounds. Anytime. Okay, I want, with the risk of really boring you, I thought it'd be important to show you why the funders were actually interested in this and how we pitched it and what the point of making this new type of therapeutic is. So there are some fundamental unmet clinical needs for all viruses. The first one is genetic diversity. This is a well-known figure from the CDC on annual influenza vaccine efficacy. You can see that some years flu vaccine efficacy is high, even up to 50%. But the virus mutates and there's genetic drift yearly. Right? All viruses are replicating and mutating. And because of the drift, the virus is continually changing every year, and we have to continually change the vaccine every year. And some years we don't guess well. For instance, 2008 and 2014. Often we don't guess well. We saw the same thing. We're seeing the same thing with SARS, right? With SARS updating COVID vaccines, that's mutating quite a bit faster. For some viruses, like HIV, herpes viruses, the genetic diversity is so great that we don't even have partially effective vaccines like this, and we probably won't get them. For those viruses, we have, thankfully, very effective antiretroviral drugs, but they have an entirely different set of challenges. I'll group them all under deployment or logistics. They have to be continually administered, right? So for HIV, they have to be continually administered lifelong for these patients in resource-limited settings like sub-Saharan Africa. And we have tried to deliver these drugs for over 20 years to sub-Saharan Africa, continent-wide campaigns, billions and billions of dollars. And the problem is it's very expensive and very difficult to do. Not that the drugs don't work, they work very well if you can take them, but they have to be continually administered, and that leads to basic, huge problems. In the last year that we have data available, 2019, there were over 1.7 actually million new infections, despite the drugs being available and being there. And it's a deployment issue. So what we hypothesized is, or what we wanted to see, could we develop a therapeutic that was single administration and like a vaccine. If it was single administration, it would also have to overcome this genetic diversity problem. So could we develop these single administration therapeutics that high genetic barriers to resistance? How do we do that? let's revisit that old idea of interfering particles. That might be able to give us a therapeutic, since it replicates, that has a very high barrier to resistance and with single administration. That's the kernel of the idea. Okay, here's how you do it. Here is the HIV genome. It's 9.7 kb from end to end. The 5' end is called, each end is long terminal repeat, or LTR. Those are the flanking elements, and the 5' end is the promoter, a single promoter. There are 9 genes shown in brown magenta here. I won't tell you about those yet. That promoter is very weak, but in the presence of one of the proteins, TAT, it gets transactivated and the promoter becomes very strong. We characterize this feedback loop. That's a whole different end of the lab. And so after infection of the cell, I'm showing you the viral life cycle in half a slide here. The virus integrates into the host genome pseudo-randomly. In red here is the viral integration. When TAT is present at high levels, the virus transcribes the RNAs. The squiggly red lines here are exported from the nucleus. They are the templates for the proteins, the other genes that I'm showing here, GAG, POL, ON, the purple dots. And if these RNAs are carrying this packaging signal, it's an RNA stem loop structure, they get packaged in the viral genome, sorry, in the viral capsid, the purple, and enveloped, and you produce viral particles. That's the viral life cycle in half a slide. Okay, so what was the idea for building one of these tips? It was based on the old defective interfering particle concept. You could take HIV, and the Hackett or von Magnus particle would have essentially been a mutation in GAG, right? Capsid mutation. And we said, let's take that to the extreme. Let's hypothesize that we can remove everything. And what we're left with is a short piece of RNA that has none of the genes. The only thing it has are the cis elements, the packaging element and the promoters and other required elements. We called this a TIP, or a therapeutic interfering particle. This is all in the imagination right now. What would happen is in the presence of TAT, and only in the presence of TAT would it be transactivated, the virus biopromoter would activate and it could be expressed. If we built it properly, we could transduce the cell. The viral RNAs that are produced, or the TIP RNAs that are produced, would outnumber the wild type RNAs. Each of them would compete, but this population would stoichiometrically outcompete the wild type. The reason we could make more of them is because it's shorter and we remove splicing. I get into the details there. And what you'd have coming out of the cell would be these pseudotyped particles that phenotypically look like HIV because they have none of their own genes. So the envelope proteins, the capsid, were all HIV, but genotypically they're carrying the interfering or therapeutic RNA. Okay. And the reason it would be therapeutic is because it would lower the viral load by multiple logs and lower the burst size or the amount of virus coming out of each cell. That's this dotted arrow right here. So the X is lowered. Go on and transduce the next cell. And by lowering the viral load, you also reduce the transmission because we know first of all, we know the single best determinant still of transmission and of progression to disease is the amount of virus in the blood, the viral load. So you would reduce those, you would reduce both transmission and disease progression. Any questions? Yes, sir.
Can a T cell be infected sequentially by multiple viruses?
Good question. For HIV, no. There's something called superinfection exclusion. This gene, NAF, downregulates the receptor on the surface that the virus uses to enter. So for HIV, there's very little, if any, superinfection. Once in a while you get it, maybe under 10% of infections.
So when they burst out, some of those red capsules, HIV, I mean, they will stochastically infect some T cells for new ones. Those cells will only have the red one.
Right, right. That's these cells right here.
In that case,
that's these cells, right?
this strategy would not work for those particles.
But these cells, no, but these are short-lived. They last about 40 hours before they die.
Then they produce many.
They produce many reds, yes. These ones produce many reds. But what we're going for is producing this population, so the therapeutically transduced population. Yeah, you can't save an infected cell. Even antiretroviral therapy doesn't save an infected cell. Protease inhibitors allow the virus to burst the cell and kill the cell, but the virus that's produced is a dud. The protease has not been effective, and therefore the viral particles that come out are non-infectious. So this is how drugs work already.
So the goal is to infect with the blue particle as many T cells as possible before the red ones have a chance to infect.
I will almost, yes, almost, and I'll show you the R not greater than 1 concept. You only have to get 1 cell. 1 cell. You only need 1 of these to start off. Sorry, Richard.
Why didn't you build your tips with NEF expressed so that it would inhibit 2 ways, outcompete plus downregulate the receptor?
Good question. If we downregulate the receptor, these guys can't be super infected by HIV, which is what we need. That's one answer. No, we want if we get the tip in, this check mark is the tip. The X, the HIV, can't super infect. This is actually the next slide will show you. Let me go to the next slide because it will give. I think it'll answer everyone's questions. I know there's a question, but let me go to the next slide. Okay, so level of virus determines transmission and progression to disease. If we lower viral level, we stave off or at least reduce both of those. Okay, so the theoretical requisites for a tip. In this case, HIV, the virus is in red. The tip is in blue. So the canonical or classical von Magnus dips have to satisfy 2 criteria. The first is conditional replication. The dip can transduce a cell, get in. We don't say infect because infection usually implies replication. So transduces a cell, but it doesn't do anything. It requires the wild type virus, in this case HIV, to superinfect that cell in order to replicate. Provides all the replication machinery in trans. Criteria 1. Okay? Criteria 2 is it has to actually do something, has to interfere. The wild type virus infects the cell, and produces some burst size, some number of viral progeny, infectious progeny. If that wild type virus infects a dip transduced cell, it produces less viral progeny. It interferes in some way. Okay? And we added a 3rd criteria. We said quantitative criteria that the R naught had to be greater than 1. The mobilization of this dip had to be greater than 1. This is essentially a bifurcation parameter, a fold bifurcation, allows the dip to have a steady state, a sustained steady state, and not be extinguished by the infection. It's what allows it to be single administration and resistant, and then have a high barrier resistance. Yes.
HIV is transmitted between people. If someone has tip intervention, can that spread between people as well?
Ideally, so this is a hot button topic. I think that's a positive. You need that for a therapeutic that's going to be a population level therapeutic. All of my collaborators say, don't talk about that. Like, let's. But I think it's super important to. Otherwise, you're fighting fire with something else. This is spreading. And you're fighting with a static chemical that is not spreading. That's a mismatch. And that's why we have most of the problems we have. That's why we have a resistance problem. That's why we have a deployment problem, is the mismatch between chemistry and biology. Biology replicates, therefore it mutates. The kernel of mutation is replication. Chemistry can't do either of those 2 things because it can't replicate. And so we have biodistribution problems at multiple scales. I usually talk about that, but I went this way instead. Replication is the key. And okay, so R naught, just for the. Sorry. Say that again.
So the virus your MS, the
Particle.
I think the line deletion inject the tap, we use that in possible.
I'm not sure. I mean, if you add TAT, you can activate the tip, but it won't produce capsid and all the other things. You'll just turn on the RNA.
It's very specific.
It needs all of the componentry, it needs not just TAT. It needs capsid, GAG, it needs POL, it needs NEF, it needs, well, it doesn't, yeah, it needs NEF. It needs every gene, because we cut out every gene.
Signal is that antivirals would activate that.
You've intuited the next slide also, so I'll show you that. Yes.
There's a risk, though, because the provirus inserts randomly in
It
the
does.
genome, and you're going to be inducing mutations in a very large population of lymphocytes and possibly cause cancer.
Good point. Okay, so I usually save this till the end. This has been, but we can go into it now. We're not going to finish, but historically, the idea of insertional mutagenesis and genotoxicity has been a major concern. In fact, every gene therapy trial in the world was stopped in early 2000s because of a retroviral gene therapy in stem cells in patients that induced a lymphoma, leukemia. Since then, the gene therapy field has done toxicology and understood what it takes, and CAR T cells are essentially exactly this, right? CAR T cells are a lentiviral gene therapy integrated into a CD4 T cell, or CD8 T cell, or CD4 T cell. And Carl June has now thousands of patient years of data showing there's no evidence at all of a lymphoproliferative event of a leukemia or lymphoma. Just last year, there was about a year and a half ago now, there was a notice from Peter Marks and the FDA that there were a number of patients, 19 patients with secondary cancers in CAR T cell treated individuals. And this caused the problem, potentially. Only 3 of those patients could you actually maybe tie it back to the CAR T cell, but 19 patients. What many people have noted is that these patients, there was a denominator. There are 27,000 people who've been treated with CAR T cells. Many of them, if not all of them, had a pre-chemotherapy. The secondary cancer rate for these cancers is 10%, between 9 and 18%. Let's just call it 10%. There should be 2,700 secondary cancers. There are 19. So that's the...
So you'll be using lentivirus.
Use the lentivirus. Yeah, and there's no evidence of genotoxicity. That's a long-winded answer. I have a longer-winded answer too. But there's no evidence of genotoxicity in T cells, and now in many other cells. The original data was a retroviral vector in stem cells or bone marrow transplant cells. Yeah. Okay, so R0 is, the definition of R0 is, I didn't go over, is basically the number of progeny infections from a single infected individual, assuming the entire population is susceptible. So if it's greater than 1, the infection spreads. If it's less than 1, the infection extinguishes. Okay, that was the key.
Just a question. You said that not super infection, right?
Yep.
So essentially, this would only work on T cells that have never seen infection, right? So do you need R in order to like as a pre-treatment, in order to like?
Good question. So two answers. These cells that are HIV infected live for 40 hours. They're such a small. They're so short-lived. You're unlikely to get them anyway. We're not going after them. We're going after these cells that have never seen, and that's the larger population. Even in an HIV infected individual, this population is 0.1% of these cells, if not less. So it's a very small subset. And even in active replication, when you have a set point over here, a very small subset are actively infected.
The tip infection rate is so high that it like.
It would, yeah, it would trend. This population would be high enough that the R0 would be greater than 1. That's the idea, and that's why you only have to get 1, theoretically. We think you have to get more, but in theory, 1's enough if your R0's greater than 1. So, okay, now I have like 10 minutes left. I think I should just jump forward. Let's see here. Okay, I'm not gonna go over the projected impact. Question.
Why?
We'll go.
Why it's like, because I know there are.
I have to go through the molecular biology for that one, but yeah. In essence, there needs to be a very specific mutation in HIV for this to happen. In order to find an R0 greater than 1, there have to be multiple mutations with a piece in the middle that is retained. And so it's some acrobatic recombination that needs to happen. That's why we don't think it happens.
Like integration, there's so many dysfunctional integrations.
Right, they are, but their R0 is not greater than 1. Yeah, you need a very specific position to be maintained. That's what we found. All of them poop out. Yeah. Okay, I will do one last slide. Because I'll go the whole time because we're just taking time. we couldn't build this for 20 years. This was just in the computer, just in the imagination. It was a beautiful concept, but it was impossible to engineer. And the only thing we could do was mathematically model it, which is weird because usually modelers will help experimentalists by making mathematical models to help the experimentalists design and interpret their experiments. In this case, this thing didn't exist and we were modeling it. It was weird, right? So here's one of the models, and this is all we had for many, many years. This is an epidemiological model of prevalence of HIV in Malawi and sub-Saharan Africa. It's a UNAIDS model from an antenatal clinic data set, women coming into clinic, boots on the ground epidemiology, and you have prevalence of HIV in that population. It was 28% in the early 2000s, and it steady stated. It was believed that nothing would, if no treatment, that it would stay at that level, and that's been about true. We looked at antiretroviral therapy. Many other groups looked at antiretroviral therapy as well. What effect that would have if you could get antiretroviral therapy to 75% of the population, and we know it's 99% effective or more. And it was sobering. It would reduce the prevalence down to about 20%. The reason is attrition. You can't continually give these drugs because people just, even in a clinical trial in Africa, come off their drugs in about 2 years. We looked at vaccines, even a 30% protective vaccine or even a 50% protective vaccine, similarly sobering. And the reason is partially protective vaccines, when you have an epidemic that's driven by high-risk individuals, multiple exposures, it's quickly overcome. Okay, we put tips into this model. Even though we couldn't really build them, we could build something in cell culture that we thought partially worked. And we got this very dramatic result where the tips could reduce viral load at the population level. Even if they only partially reduced viral load by half a log, would dramatically reduce the prevalence of HIV in the population. This is purely computational. This 0.5 logs is what we could build in cell culture, but that's all we could do. And we said we can build something that reduces by maybe a log. This was what drove us. This is all we had for 15 years. I'm going to skip this even though someone asked about it. We'll come back to it. It's the evolution argument I'll come back to because I want to get to the data in 5 minutes, at least show you that we built it and what we have. Okay, I'm going to skip the screening. How do I do this? Okay, so we are eventually able to build one. I'm not going to show you how. We were able to build the tip through various screening approaches and years of trial and error. And we put a blue fluorescent protein in the tip. We have a red fluorescent protein in the NEF reading frame of HIV. We transduce all of the cells here. This is a microscopy movie. Transduce all the cells. Okay, so that went a little too fast. Okay, so this is a 40-hour microscopy movie, what a confocal movie. I've stopped it at 15 hours. You can see the cells are HIV infected. Red is showing up. The NEF, the red is actually nuclearly localized. And you can start to see a little bit of blue coming up, activated, right? Let the movie go past 15 hours. So all the cells were transduced with the tip and then super infected with HIV, right? And then here's what happens. The tip takes over, is expressed. You can take the supernatant off of these cells and do a serial passage experiment where by flow, you look at percent of HIV infected cells. HIV destroys the culture in about 21 days. In this particular setting, it's a bioreactor, whereas the tip transduced population, even though HIV is present, is maintained at low levels. So we can protect the culture from dying. Okay, that's in cell culture, great. But the real gold standard is, can you do this in an animal? So we built a version of this tip in SIV and went into monkeys, and the leading model is rhesus macaques. It is an Indian origin non-human primate, and you can give it an SIV from an African primate, and it gets an AIDS-like disease. And so we chose the most pathogenic model that is in the literature with Nancy. These animals are infants that are given SIV, and this is the viral load of SIV. The virus goes to very high, 10 to the 8th copies per mil, and doesn't come off the peak. It sort of stays at the peak. These animals succumb to their disease in about 17 weeks. Okay, they quickly die, and that's why we chose this model, because of how quickly these animals succumb to their disease and reach clinical endpoint. Remember, if they went down to lower levels, it would take much longer for them to reach disease. We gave them a single administration of the TIPS, lentiviral vector IV, intravenous, either 24 hours before infection, 4 days after, or 10 days after. All the data looked pretty similar. And this was the viral load data in those monkeys. Went down by about 4 logs. These animals lived, as far as we could tell, they were healthy. We sacrificed them at around 30 weeks for tissue and analysis, and FDA wanted those things. And then remember, the hypothesis is that it's a single administration. We should be able to see the tip. Even though we gave it once, it should be propagated. We should be able to see it. And so Fatima developed a sensitive ddPCR assay, and we were able to, even though the tips were given a single time, 30 weeks later, you can still pick up the tip genome. You can do the calculations for dilution factor of how diluted that would be 30 weeks later in animal. It's right. The numbers are like astronomical. They're silly. They don't even make sense. Okay. So only way you get this is with replication.
Have you tried multiple doses over time? You must have.
We tried it. We didn't see a difference. We tried another dose in some of the animals over here. No difference. So we also did this in patient cells. Well, for here, first is the survival data. This is the Kaplan-Meier survival data. This is, yeah.
Steady state should be, in the end, if you had infinite time, it should go back to 0, right? Or close to 0?
The steady state should be maintained.
Or at a certain level.
It should stay.
But it's still going down a little bit, right?
Yeah, this one's going down a little bit. Not sure why. If you look at individual animals, they're pretty stable, but this one sort of dominates your eyes.
And
Yeah.
that load is 10 to the power 4.
About 10 to the power 4.
But that is from what you know from patient samples, it's okay.
Well, if you do the calculation for humans, which we did, humans don't have a viral load of 10 to the 8th. They have a viral load of about 10 to the 5th, 10 to the 4th. So they start over here.
So that's okay.
So 3 logs below, if you can go down even 2 logs below 10 to the 4th, you're 10 to the 2. 10 to the 3 is the WHO, the World Health Organization, threshold for viral transmission. So if you can get below 10 to the 3, you stop transmission. So we can get 4 logs. That's a big deal. In humans, we predict we can get 3 logs, which will be quite good. Yeah.
Okay.
And I'm almost finished.
I think you can take 5 minutes more because we started at 5 minutes late.
I can keep on going. I can go back to the.
Something interesting to discuss.
Yeah. What I'll show you. So this is all in a single molecular clone of the virus that you give the animal. It's not a diverse population that you gave the animal. A molecular clone that was developed in the lab that was cloned. So there's no diversity to begin with in the animal. There's quickly there's diversity, but what about in patient cells, in patients, which have a lot of diversity? So we were able to get patient cells. I told you about this already. We were able to get patient cells from a cohort of patients, injection drug users who have been drug naive or treatment naive. And we could do a viral outgrowth assay from these patients and treat their cells in culture with TIPS, and we would see a reduction in viral load, basically arguing that it's not just a molecular clone, it's a diverse human virus population can also be knocked down by the TIPS. So some preliminary evidence. We took all of this to the FDA, and we now have this clinical trial that's funded and is in IND stage. So, okay, what do you guys want to see? I can show you SARS-CoV-2 TIPS that actually came in a quirk of publishing, were published first, even though the HIV was discovered first. I can show you the molecular data, which I suspect this crowd is not as interested in. I can show you the evolution argument, which people are usually very interested in. What do you guys want in the last 5 minutes?
Evolution.
I figured you want evolution. Of course.
I seem to recall retrovirus genomes have to be a certain length to be properly packaged. And the way you've drawn it is your little blue fella is much shorter in length. But is that just sort of like a figurative?
No.
Do you engineer it so?
It's shorter. It's shorter. It's less than half.
Oh.
We have less than half.
But
and
I'm
we
remembering.
Retroviruses are much shorter to begin with than lentiviruses. Lentiviruses are 9 to 10 kb or longer. Retroviruses are typically, 6 or so. So they're already much shorter. But if we go too low, too small, we lose certain cis elements that we need for packaging. That's the part I didn't go through. We're not sure that there's an actual length requirement. For other viruses, like the Sabroca viruses, the SARS-CoV-2 and those viruses, there is a length requirement. But for HIV, we haven't found one. We just need certain elements. Yeah. So there is a threshold. If you don't have those elements, I mean, you can't go below a certain length without deleting one of those elements. But if you have that length, it seems to be sufficient.
I was just going to ask, can you optimize these tip cells in other ways? Maybe your LTR, you could do some base editing screen or something which identifies the stat feedback much stronger.
We looked for things like that where we could try and optimize the LTR, and this is part of the evolution argument, right? So it turns out that this virus is so optimally evolved that you can't do better. And that's been done specifically for the packaging signal. Ben Burkhout in the Netherlands in the 1990s tried to mutate both the tar loop and the packaging signal to try and find optimal packaging variants and optimal tar loops. The wild type is the optimal. Yeah, so makes sense, right? Yeah. Yeah.
That's a question. Okay, so you're perturbing the system, right? And it has this great kind of therapeutic. But what are the costs of doing this, right? Are there other, like, you're pushing this kind of biological system out of equilibrium, right? And how should I think about it?
Here's the cost. You're never going to cure. Drugs theoretically have the possibility of being curative, right? In theory, you can cure a population. You could end the epidemic in theory. But in practice, you can't do it because of latency, because of deployment issues. Here, you have no theoretical chance, none, of curing either the patient or the population. What you do trade for that is the practical possibility of controlling at very low levels down to 1%. That's the trade-off.
Okay, so the additive then, how can I be sure that it's not affecting other physiological processes like this, right?
Very likely it's going to be affecting other physiological processes. This is an HIV infection to begin with. So you're not going to give it to healthy people, you're going to give it to HIV-infected individuals. In individuals who don't have HIV, it's just going to sit there as a gene therapeutic agent, as an integration. And I wouldn't give it to healthy individuals. Every therapeutic has an off-target that has unintended consequences. We don't know what these are until we test them in humans. In monkeys, we couldn't find anything. Yeah. Okay, the evolution argument. Okay, so this is usually generates a lot of discussion. One, let's start off with current therapeutics. Viruses, because especially retroviruses, mutate so rapidly, they produce what we typically refer to as a quasi-species or swarm. Manfred Eigen developed this theory in the 1970s. He won the Nobel Prize for something completely different, but this is Manfred Eigen's quasi-species theory. In HIV, the viruses are, this polymerase or reverse transcriptase is so error-prone that every single replication event, there's a mistake made. The error rate is about 10 to the fourth. They're actually a little less, a little more than 10 to the fourth. It's in the 10 to the thirds in some measurements. The virus genome is 10 to the fourth bases. Every single time you replicate, you made at least one error. The virus is replicating on 100 million times a day in an individual. You're making every possible single point mutation many times over every day, and a good fraction of all double point mutations. Okay, so that leads to this quasi-species of genotypes. You have all these different genotypes. And the dominant one is the fastest replicating one. That's the species that is mostly, it's dominant by a lot. It replicates more quickly. What happens when you give drugs? Well, the drugs were developed against that wild type, usually in the laboratory. So they knock down the wild type, but often especially for a monotherapy and for dual therapies, there'll be a variant somewhere else in the population that just pops up. And this happens even in triple drug therapies now, we know. So this is the problem with monotherapies and why and it's a problem in cancers as well, why we give multiple drugs. So for TIPS, the idea is a little different. Remember, TIPS are replicating with the same reverse transcriptase, same error rate. So they're producing a quasi-species. They will produce a quasi-species. And the idea is, we don't even know if there's a variant that can arise that can resist or that can escape the TIP. But if there were, we believe there'd be a reciprocal mutant from the TIP that would compete with the new resistant mutant. So, and then here's an example of that. Let's say HIV mutates its packaging signal so that it's different and HIV can no longer, and TIP can no longer be packaged by the wild type, right? Well, it can do that, but it has to reciprocally mutate its GAG protein, the capsid protein, to recognize that new packaging signal. If it's just different enough that the GAG doesn't recognize it, it won't help it. It's got to mutate both of these, this and this. This guy's mutating at the same rate. And so it's going to already have that mutation as well, right? So and this is 2 mutations that have to happen versus only 1 mutation because this doesn't have GAG in it. So it's always 2 against 1. Any idea, any mutational mechanism that you can come up with is always going to be 2 against 1. There is. Can do recombination. Not sure how that helps other than, well, one thing that could happen. Go ahead. It could. Usually, I talk about that in the context of toxicology or unintended consequences. Let's say you create, people say, well, what if the TIP starts mutating and acquiring things to become a supervirus? Like, right? What if you create a supervirus and you make things worse? So how would that happen? Well, it would start to grab things. Well, HIV can't really grab anything. There's nothing to grab. But the TIP could start grabbing things. Let's say it grabs everything, right? It grabs GAG. Great, it has GAG. Let's say it grabs Paul. In order to become a virus, it has to grab everything. That can only happen in the presence of the wild-type virus. It grabs everything. It becomes the wild-type virus. We already deal with that. It's called the loss of efficacy for a drug. When the TIP becomes HIV, it's not effective anymore. That only happens in the presence of the wild-type virus. Okay.
What if were to mutate in that?
Mm-hmm.
Turn off superfractions, and then you would have more integrations of HIV that could like overpower the TIPs.
You could. These cells still last for some small amount of time. So it's not these cells that we're going after. It would have to happen over here. And it's so hard to get superinfection in the first place that it's a small subset, small percentage. Yeah.
Is it so hard as Metzger, or is it like in general?
NEF is one mechanism. The other mechanism is the short lifetime. Both of them, yeah. No, it's the virus kills the cell in 40 hours and NEF downregulates. Both of them are at play. Yeah. Yeah. Yeah, you're already going to get multiple integrations of the TIP probably, we believe. 2 to 3 is what we believe. But yeah. Yeah, and even if you have multiple copies, we tested that. It doesn't help that much because even a single copy, we've removed all the splicing. So you're getting 10 times as much TIP compared to 1 HIV. Yeah. And I guess I'll went over time. I guess I'll leave it there. Thank you guys.